Bismillahirrahmanirrahim. Assalamu alaikum. How are you, dear students? I hope everyone is fine and be healthy. Today we are going to study one of the most important topic of the 10th class, that is enzyme activity. We have studied in the previous lesson enzymes and metabolism. So our today's topic is belong to the enzymes topic. So we are coming here to the enzyme activity. What do we have in enzyme activity? We have temperature. So we can say effect of temperature on enzyme activity. The temperature which allows the highest enzyme activity is called the optimum temperature. What do we have here? We have temperature here. So which type of temperature is the optimum temperature? The temperature in which the highest enzyme activity is happen. So we can say that this type of temperature is named as or is called optimum temperature. We have different enzymes. So different enzymes may have different optimum temperatures. In our body, there are many types of different enzymes and we have different substrates. For each substrate we have its own enzyme. Now each enzyme has its own optimum temperature. So simply we can say that different enzymes may have different optimum temperature. The optimum temperature is not constant for all types of enzymes. At high temperature, the shape of active site is changed. This process is called, or this process is known as denaturation. If you remember from the previous lesson, what did we study? We have studied that we have, for example, this enzyme. What is it? It is a type of enzyme. Just suppose that it is the shape of enzyme. What do we have here? This part is the active site of the enzyme. Active site. Now what do we have here? We have a substrate. Now the substrate is combining with the enzyme. After that we can get an enzyme substrate complex. Now this active site is affecting on the substrate. At high temperature, when we have a high temperature, not the optimum or constant temperature, what is happening? The shape of the active site, this side, the active site shape is changed. How does it change? Like this. For example, now this substrate cannot combine with this enzyme. So what can we get? We cannot get an enzyme substrate complex. Why? Because of the high temperature. The high temperature changed the active site, and when the active site is changed, so the substrate cannot be affected by this enzyme. So we can say this process is called or is known as denaturation. The optimum temperature of enzyme is in human body is not 37 Celsius degree. It is our body temperature. Our body temperature, normal body temperature is 37 Celsius degree. Now we cannot say that 37 Celsius of the degree is not optimum temperature for enzymes. Actually it is the temperature of our body. If we look to this graph, what do we have here? We have at this part the temperature which we can measure it with Celsius, and at that part we have the enzyme activity. Now what is happening? We have here the Celsius degrees which are starting from -20, -10, 0, 10, 20, 30, 40 and 50. These are the Celsius degree of the temperature. Now what is happening? When the temperature is minus 20, the activation rate of the enzyme or reaction rate of the enzyme is very decreased. When the temperature is decreasing, when it is increasing, when the temperature is increasing, at that time the activation rate is also decreasing. And we have an optimum temperature for this enzyme activation between the 20 and 40 centigrade Celsius or Celsius degree. So this graph is belong to the activity of the enzyme which is related to the temperature. So we can say we have different optimum temperatures for different enzymes. And the second thing which we have in enzyme activity, that is the pH. What does it mean? The pH is abbreviated like power of hydrogen. Power of hydrogen. With power of hydrogen, we can find the acidic and the basic state of some materials. Now we are going to study the pH, that how can the pH affects the enzyme activity. In the previous part, we mentioned that the temperature which affect on the enzyme activity. In this part, we are discussing the pH which affects on enzyme activity. So the pH value at which the enzyme has the highest activity is called the optimum pH. At that part we had temperature, but in this part what do we have? We have the pH. So the pH value at which enzyme has highest activity, this pH is named as or is called optimum pH. pH of medium, for example, we have a medium here, higher or lower than the optimum pH can denaturate the enzyme activity. If we don't have an optimum temperature, an optimum pH for the enzyme activity, in that case the enzyme cannot work properly. Now look here in this graph, what do we have here? We have at this part the pH, and at this part we have enzyme reaction rate. The neutral state of the pH, in which some materials are in the neutral state, that is the pH of 7. From 5 to 7, this part is the neutral part. When we come below the 5, or when the pH is decreased from 7 to 1, so we can say this part is the acidic part. The material have acidic state. When the pH is higher than the 7, in that case we can say these materials have the basic state. Now the optimum and a good pH for the enzymes is from 5 to 7 to 8, we can say. So this part is the neutral part. At this part, what can we see? Here we can see the neutral state. At this state, the enzyme can work properly. So it is the optimum pH for the enzyme activity. Like temperature, also pH is the same. For many enzymes, we have different types of optimum pH. Now look to this graph. Different enzymes may have different optimum pHs. What can we see here? We have at this part the pH level, and here we have the enzyme activity. If we look, we can give some examples of the enzymes like pepsin. Pepsin is a type of enzyme which is found in our stomach. Now, the optimum pH for the pepsin is 2 or 2.5, we can say. So the pepsin can be activated in which type of pH or which type of state? Acidic state. Why? Because from 2 to 3, here we have the lower pH that is lower than 6. It is not in neutral state. It is in acidic state. So pepsin can be activated, or we can say the optimum pH for the pepsin is from 2 to 3. And we have other example. Most enzymes can be activated or they have the optimum pH from 6 to 12. Most enzymes. For most enzymes, the optimum pH is from 6 to 10, we can say. And another example for basic. For basic state or alkylic state, what do we have here? We have an example of enzyme that is lipase enzyme. And this lipase enzyme is produced by our pancreas. So we can say pancreatic lipase. For pancreatic lipase, the optimum pH for this type of enzyme is from 10 to 12, we can say. So the pancreatic lipase can be activated in a basic state. So this state is a proper state, or we can say the optimum pH for the pancreatic lipase. So it was our today's topic. Inshallah, in next video, we will study the types of reactions. Types of reactions. If you have got any question, you can ask in WhatsApp group. Have a good time.